Hello, welcome to the next session on pancreas anatomy and physiology. So today I'll be talking a little bit about the pancreas itself, and in future sessions we'll go through clinical presentations of where the pancreas goes wrong. For example, the most common one is pancreatitis. So let's go to our first one. So we'll start off with the anatomy. So what is the pancreas? So it's an oblong shaped organ. We can see it looks just like this, and it has four general parts, five actually, but you can't actually see the fifth one. So here it says the unsnit process. You've got the head of the pancreas, the neck, the body, and the tail, and this all makes up the pancreas. So in our gut, we've got a peritoneum, which is like a covering for our gut's organs. Some organs are inside, some organs are outside. The pancreas is generally a retroperitoneal, which means it sits behind the peritoneal cavity, except for the tail, which is actually intraperitoneal, which sits inside. So that's a brief anatomy in terms of its different types of sections. And you can see that it has quite a direct relationship with a lot of the organs in the gut. It's quite proximal, close to the kidneys. It's got the duodenum that sits quite snugly with the head and the unsnit process. And you've got the aorta, the large vessels in the body running posteriorly or behind it. On the right, we've got another diagram, and it kind of represents the inside of the pancreas and what actually goes on. So as we know, the pancreas is very important in the body for two kind of main functions. One function is digestion, which is it secretes a lot of the enzymes that are important for digestion. And the second one is that it's very important for controlling a lot of our hormones, in particular, blood glucose levels in our body. So here in terms, we can really clearly see all the ductal system that's going to be important for our enzymes. So on top of here, it's not shown here, but here it's the liver. And here there's the ductal system. You've got the right hepatic duct, the left hepatic duct joins down to the common hepatic duct, and it comes down all the way here and exits into our duodenum. And here you've got the pancreas. So you got the main pancreatic duct, and the main pancreatic duct kind of joins in with the common hepatic duct and exits out right here. And then we've got an accessory pancreatic duct. So an accessory pancreatic duct also exits out here. Oops, let's go backwards. And the accessory pancreatic duct exits out here into the minor duodenal papilla, and the big one is the major duodenal papilla. So here we've got the duodenum. It kind of clearly represents our four parts of the duodenum, the superior part, and then we've got the descending, the inferior, and the ascending part of the duodenum. So in terms of the pancreas, important to remember our ducts, our two main ducts, and where it exits. And then it's also important to remember the common hepatic duct. In terms of papilla, papilla in Latin is nipple, and it kind of resembles its shape as well, and hence the naming of the structure. So the next part is, we're going to look closely. So we're going to zoom in on where that kind of exits into the duodenum. So when we zoom in, we can see here's the common hepatic duct, as we can see here, the common hepatic duct, and it joins in with the pancreatic duct over here. And there's some sphincters. So sphincters are responsible for closing and opening, because we can't have it open all the time. All the enzymes and stuff that it's secreted by the pancreas will be constantly going into the duodenum. So we need something like a valve system. So we've got sphincters in place. So we've got the common bile sphincter, the pancreatic sphincter, and the ampulla sphincter. So the section where these two join together, it's called the ampulla, and hence ampulla sphincter. All of these sphincters all together make up something called the sphincter of Oddi. So why is it called ampulla? Ampulla is Latin for flask. So we can see here, it kind of looks like, if you rotate this 90 degrees anti-clockwise, it kind of resembles this ampulla structure over here. It's like the top of the flask in a way. Alrighty, so the next part is its relationship with the organs. So as we can see, it fits in well with the duodenum, especially the head and the unsnit process of the pancreas. It's also related directly to the stomach, but it's separated by something called the lesser sac, as we can see here. So the lesser sac is this area here that can be accessible through this omental foramen, as mentioned over here. It's also attached to the transverse mesencolon. And also we've got the common bile duct, which actually runs through it, which you can't actually see and appreciate in this diagram right here. We've seen it in the previous slides, but we've got the large major vessels running posteriorly behind the pancreas as well. So a lot of important organs that surround the pancreas. So here's a quote: "One can only understand the essence of things when one knows their origin and development." And this is by the famous Alexander Oparin. He's a famous Soviet biochemist, I believe, and he was well notable for his theories on the origin of the Earth and its life. So he kind of he made a theory that suggested that Earth, when it started off, had primitive oceans, which there was, he framed the term primordial soup, I believe, or primitive soup, and it consisted of chemicals and compounds which would later reform and assemble to the life we know right now. So this is important in medicine because it was really important to know how everything starts off with. So when we think about the embryology, or how the pancreas started off with, here we've got the duodenum, and here's the foregut as it starts off. Here we've got the gallbladder, the cystic duct, and the common bile duct. We've got the ventral bud and the dorsal bud. So there's two sides. The ventral bud kind of swings around and connects with the dorsal bud, and it fuses to form what we know as our pancreas. So here we can really appreciate the pancreatic duct and the accessory duct that we talked about before. So our major papilla and the papilla minor. And there are sometimes abnormalities in this process where the process of fusion. When it fails to fuse properly, and has a ring-like structure, it's called an annular pancreas. This is a rare condition that's found, and it can present with symptoms of pancreatitis or even incomplete bowel obstruction due to its obstruction of the duodenum. And usually surgical correction is required. So the next one we want to talk about is the function of the pancreas. So there's two main functions, as we talked about: the digestive side, the exocrine function, and the endocrine function. So exocrine means it's producing things into ductal systems. So here our pancreas is producing digestive zymogens, and these are enzymes which will later be activated in the duodenum. So our common ones are amylase, lipase, proteases. And these are produced by our acinar cells. The second function is the endocrine function. So this will be probably a separate session, but we'll briefly touch on them, that these are commonly found in little sections in the pancreas, and they're called islets of Langerhans cells. Here we've got particular cells: alpha cells, beta cells, and delta cells. There are a few others as well. The beta cells are responsible for producing insulin. Alpha cells are responsible for producing glucagon, and delta cells are responsible for producing somatostatin. And we'll go into more depth about the functions of these hormones in the future sessions. So let's talk about the exocrine function, the digestive side of the pancreas. So it's responsible for secreting pro-digestive enzymes. And so it's first secreted as zymogens. So here it's packaged and produced as inactive enzymes, which will later be activated to its active form. In brackets, it kind of suggests what type of food it's going to be digesting. So trypsinogen will be activated to trypsin, which will later digest protein. We've got proelastase, which will then be activated to elastase, which will digest protein. We've also got prolipase, which will be digested to lipase, and then which will later digest fat. Zymogens are packed in granules to avoid autodigestion. So it's really important that these enzymes are packaged and transported carefully from the pancreas into the gut, because we don't want the enzymes activating and attacking and digesting the pancreas, which is the term autodigestion. And amylase is usually released in its final form. The pancreas can also produce bicarbonate-rich fluid via this hormone secretin. And this is it's a protective function of the pancreas. And we'll discuss this in the future slides. So let's go to the next slide. So here we can see the acinar cells, and they are responsible for producing the digestive enzymes. Then here we've got the ductal cells, the pancreatic ductal epithelial cells, which are responsible for producing bicarbonate-rich solution. This is a protective function because we don't want these acidic solution and secretions to damage the pancreas. So here we can see this area is quite acidic, and it turns to be less acidic while it passes through the ductal system. And here we can see that the acinar cells are arranged in a lobular structure, and it's connected via intralobular ducts, which then drains into our main pancreatic duct. So here we can see that quite clearly, which is really good. So how do we, it's a really big concern that the pancreatic enzymes could autodigest the pancreas. So how do we prevent this autodigestion process? So there's four kind of common ways. We can see that zymogens are made in an inactive form and then transported and then later activated in the gut. Also, the zymogens are packaged in granules, which is then transported into the gut. It's also co-packaged with a trypsin inhibitor to make sure that trypsin doesn't activate early on in the process. And there's a geographical separation of the zymogen release and activation. So here the zymogens are released in the pancreas here, and then zymogens are activated in the gut here. So here it clearly sees the process. So the main key activator for this whole process is trypsinogen. Trypsinogen is secreted into the gut and then activated by the brush border enterokinase into trypsin. Trypsin then later activates all our other enzymes into its active form. So trypsin will activate proelastase to elastase and so on, and become active enzymes. These enzymes can then act on the food in our gut and digest the food for absorption. So how is this process all regulated? So here we can see whenever this is a cephalic and gastric phase. So cephalic phase is when it's kind of the pre-meal period where you see the food, you've tasted the food, and you're smelling the food. These signals go to the brain, and then it's transmitted through and controlled through the vagal system. The food in the gut, food in the stomach also goes through that same process, and it's mediated by the vagus nerve. So there's two kind of processes. The vagus nerve can then go on to our G cells that are located in the stomach. So here we've got gastrin, gastrin producing peptide, gastrin releasing peptide, and it acts on the G cells to release gastrin. Gastrin then acts on CCK receptors to release some of these enzymes for digestion. The vagus nerve doesn't actually have to also go through this G cell and gastrin pathway. It can actually directly act on the acinar cells through the M3 receptors, releasing ACH to then release these digestive enzymes for digestion. Also, it can also act directly onto these ductal cells to release bicarbonate as well, preparing our gut for that process when that food arrives in our duodenum. So the next stage is when the food's actually in our duodenum or intestine. So here we can see there's fat and protein. So fat and protein can innervate the vagus nerve and again directly interact with our pancreatic cells to release enzymes, and also directly act on our pancreatic ductal cells to release our bicarbonate solution to make sure that there is a protection. It's a protective function of the pancreas, as we've mentioned. Then, because of that acidic content, it can activate S cells in our duodenum to release secretin. Secretin can then encourage more release of this bicarbonate production. So it kind of neutralizes this acidic content in the gut. Furthermore, protein and lipids can be detected by eye cells and they release CCK, which then acts on our pancreatic duct, pancreatic acinar cells to release enzymes. So it's saying, hey, there's still food in our duodenum. We still need more enzymes to digest this food. So here we've just talked about CCK and secretin, both released from the duodenum. Secretin is in response to acid, while CCK is in response to fat and protein. So here's some specific roles. I've listed some more roles of secretin. It inhibits gastric emptying. It inhibits gastric secretion. It induces that secretion of bicarbonate and stimulates secretion of bicarbonate-rich bile as well in the liver. So all of these you can see are responsible for kind of neutralizing or reducing the acid in our gut. The role of CCK inhibits motility and secretion, stimulates more enzyme release from our acinar cells, and can induce contraction of the gallbladder and relaxation of the sphincter of Oddi. So as you can see, CCK's main role is cholecystokinin, main role is to induce more digestion of the fats and protein that is still remaining in the gut. So last thing I want to do is to talk about our blood supply to our pancreas. So our blood supply comes from two major arteries. So here we've got our descending aorta. So we've got our heart, it goes up and goes down to our descending aorta, and there's two big branches, our celiac trunk and our superior mesenteric artery. So let's talk about the celiac trunk first. The celiac trunk gives off a splenic artery, and here it gives off many of the branches to supply, especially the tail and body of the pancreas. The celiac trunk also gives another branch called, this one is the common hepatic artery. And from here, we've got a gastroduodenal artery that branches off and splits down into our anterior and posterior superior pancreoduodenal or pancreoduodenal arteries. So remember, there's an anterior and posterior, and this is a superior pancreoduodenal arteries. All right, so we've talked about supplying the body and tail and the superior part of this pancreas. What about the inferior part? Well, this is done by the superior mesenteric artery. It gives off two branches, the inferior, anterior, and posterior pancreoduodenal artery. Now our venous. Our venous is a little bit more simple. We've got our superior mesenteric vein and our splenic vein, which then merges together to form our portal vein. And this goes to our liver. Here we can see there are superior pancreaticoduodenal veins. And we've also got the splenic vein, which is also draining parts of the body and the tail of the pancreas. But these are two main veins that we should remember that merges to our portal vein. So that concludes the session on the anatomy and physiology of the pancreas. Put some comments down if what other topics I should cover and extensions of this area of the anatomy and physiology of pancreas. And I'll see you guys next session.